But I'll be talking to you about inflammaging, which is an important part of HIV pathogenesis in understanding its link. And when we look at HIV and aging in the U.S. and even on the global basis, we now know that we're seeing a greater age. In 2012, 55 or older accounted for 24% of the 1.2 million. We're now moving in 2017 and 2018 to over half of our population in the U.S. and even more in the global AIDS epidemic to finding people aging with HIV. And I think that's going to have a tremendous impact as the folks here today in your own practices or your research in Brazil are going to be able to see new non-AIDS comorbidities developing. We're seeing immune damage that occurs during the normal aging process in our HIV patients in an accelerated fashion. And there's now potential for this synergistic impact and interactions between the facts of HIV and aging together to see additional effects. We're also finding now the long-term effects of heart. We may, and people may talk at this meeting as well about toxicities. We were having even discussions last night in the bar about the long-term effects of heart on our populations up to 20, 30, or 40 years. And those combined with the other effects of aging will have significant impact. We're also seeing the non-AIDS comorbidities that are developing in our patient population. We've improved the life expectancy in HIV, and this has been a tremendous opportunity, as you can see here in this slide. If I can get, it doesn't show up very well. But what you can see now is that the HIV population is living longer, and we're seeing, though, that there still exists a gap. This is the life expectancies remaining. So you're going to still see a gap in the individuals. And the gap is only 6 years if the CD4 nadir is greater than 500 and there's no smoking, ethanol, or hepatitis. And I think this is something, again, populations here in Brazil and all over the world need to recognize, that many of the lifestyle things that we take for granted, like smoking or alcohol, diet, are all going to affect dramatically our HIV populations as they're aging. And within the clinic populations, you need to be counseling your patients about these aspects. And as well, we need to understand more about their impact on HIV and the aging process. We also know that if we look at C4 nadir, there's a 20-year reduction in life expectancy if the nadir is below 350. So again, getting patients into therapy early or starting patients when they're first diagnosed is critical. And as we see here, of the 20 million people on ART globally, the vast majority did start with ART at less than CD4s of 350. So that's going to have a dramatic effect then on the life expectancy, and we need to see, as we're able to treat and identify patients earlier and start them on therapy, hopefully a better impact on their life expectancy and outcomes. We're also showing that many of the morbidities are declining in the modern ART era, although some persist. And here in myocardial infarctions, we can see that there are changes, and I'll talk more about those today. Also, neurocognitive dysfunction is going down as well. And we know then that the increase as well is very much found in the virus-associated cancers. We're seeing much more even in our early HIV patients that are treated within the START trial, saw many of these AIDS-associated viral cancers developing in populations even with high CD4 counts. And then finally, what we have is really a very strong epidemic globally of chronic liver disease, and that's what we're seeing more in our patients now, is the liver is going to become a much more prominent area for study, both from non-alcoholic fatty liver disease and non-alcoholic steatohepatitis. So I think we're going to be seeing much more liver disease, and as well, the viral hepatitis are going to be important. So what do HIV infection and healthy aging have in common? Well, overall, what we can find, there are many similarities. There's really an important intersection here between the immune changes that are shared, and these include the pro-inflammatory cytokines. The one that's been most well studied is that of IL-6, and that's been associated with HIV mortality and morbidity. We're also very well of coagulation markers, and the tissue factor work and also D-dimers have been shown to be important as clinical outcome measures. And then finally, There's the pro-inflammatory monocytes that are getting more attention at the cellular level, that they're driving the age-associated morbidities and associated. We also know that there are shared immune clinical characteristics, decreased pathogen protection, and especially decreased vaccine responses. As you'll hear in the next series of 3 talks from vaccine experts, as we begin to consider the global vaccine effort in HIV, we also have to think about the aging population. What is the effect of aging on the immune response to the vaccine overall? And as we're thinking about therapeutic vaccines in our HIV population, that's another critical outcome measure. And then there are also shared immune changes, increases in senescent T cells, the CD28 negative 57 cells are expanded, and decreased in telomere length. So overall, we see this increase in cardiometabolic disease, type 2 diabetes, visceral adiposity, osteoporosis, and malignancies, all of which are now contributing to this premature aging, accelerated or accentuated aging. We're now very interested, as we move on in the AIDS epidemic, to study more about host microbiome and interactions. And in this case, there's a lot of interest from some of the original work from Jason Brenchley and Danny Dewitt, looking at the role of the gut permeability barrier, the systemic migration of gut microbial products. We now know that there's also an important role within the gut itself in terms of the gut microbiome. And we're beginning to study that extensively in a number of our clinical and experimental studies. We now recognize that these gut microbiome can be closely linked to the non-communicable diseases, so that they are the source of some of the inflammatory mediators and the metabolites, as I'll show you in the next series of slides. And if you look down here on the right-hand part of the slide on kidney, lung, metabolic, bone, cardiovascular, neurocognitive, and cancer, all of these have been associated both in the HIV and in the non-HIV world with the microbiome and outcomes. So I think now it gives us a new and potentially interesting target to look at in terms of how we intervene and deal with the microbiome. Where we've been able to focus, and some original work here from Ivan Vukovic's group at UCSF with Mike McCune and others, was this original work done in the primate model, showing that in the uninfected macaques, you have high amounts of lactobacillus, low amounts of the enzyme indoleamine 2,3-dioxygenase, and low levels of kynurenins. This is a protective effect on the host gut barrier and the gut microbiome. What happens then in either acute SIV or HIV infection, there's a decrease or loss of these important lactobacillus, increase in the IDO pathway, increase in kynurenin, leading to this local activity and inflammatory state. And we're beginning to now study this whole pathway, as you can see on the left-hand side, of tryptophan catabolism, kynurenin, and indoleamine 2,3-dioxygenase, or IDO. I might also just comment, IDO is one of the newer therapeutic approaches for cancer. There's a lot of interest now, as you know, about the checkpoint inhibitors with cancer. There's also a lot of interest in trying to block the IDO pathway, which are capable of blocking the host immune response. So we are starting to look and do those same types of studies. In our particular study design, we've used 2 large cohorts to look at the outcomes of changes in tryptophan catabolism and cardiovascular disease association. This is the Women's Interagency HIV study and the Multicenter AIDS cohort study, where we did carotid artery imaging. And in this case, we had longitudinal data on both the WISE women and the MAX men pre-development of the incident carotid artery plaque. So we were able to look and see what were the predictive measures of the tryptophan pathway. In cases here, we did repeated measures for intermedial thickness and plaques and stiffness. In this case, we were able to show very clearly that there was a loss of tryptophan here in our HIV viremic patients, and also an increase in our kynurenic acid to tryptophan ratio. There's a highly significant level of this ratio that did, as I'll show you on the next slide, predict and had a strong association with risk of carotid artery plaque. And this was a finding where tryptophan itself is protective, and that the increase in the kynurenic acid-tryptophan ratio, as noted here at the bottom of the slide, is actually affecting and has a stronger predictive value for a negative outcome in cardiovascular disease. So I think this gives us a novel new biomarker and also an interesting target to think about in terms of reversing this kynurenic acid-tryptophan ratio. I won't talk today, but there's also substantial data in HIV and neurocognitive outcomes, and a variety of groups are studying this, and the same kind of data is coming out in correlating changes in kynurenic acid and tryptophan and neurocognitive disease. So I think this provides us with the IDO pathway and an interesting new and potential target. We were also able to show that the plasma tryptophan metabolites are independently associated with risk of carotid artery plaque, and that the ratio itself was the highest level when we further adjusted for all the other inflammation and immune activation markers. So this really was an independent predictor of outcome of our more standard markers and gives us new pathways to identify and pursue in terms of pathogenesis and therapeutics. We also, in this heat map, did look at Spearman correlations with some of the inflammation and activation markers and the traditional risk factors. And what we found was the most significant here at the top part, you'll see, was soluble CD14 in the kynurenine tryptophan ratio, and then also CD4 T cell activation. So it's interestingly different here that CD4 T cell activation was the highest correlate with the increase in the actual ratio. And we're now beginning to look at that further in terms of the drivers of T cell activation and the intersection, as I'll talk about later, with this area and HIV viral reservoirs. So what we're now doing is we're further pursuing this work in terms of the gut microbiota, host metabolites, and CVD. And what we want to do now in a larger cohort study between WISE and MAX is to identify the specific gut microbiota and related gene functions that will be contributing to these metabolites. So in our study design, which we've just begun, this is under a newly funded R01 grant in the US, we're looking at 365 WISE participants and another 200 MAX. So we have a total of 500 participants in this study. We're getting fecal sample collection over time and then also doing this at home. So this is a very practical approach. So for those of you interested in doing this kind of work and could work out in the community, I think we've really developed this effectively where we can collect the stools at home and then have them sent in through use of these FTA cards, or directly by sending tubes of RNA later where the samples can be taken at home from the subjects directly. We're also then looking here, as you can see, at the gut microbiome, metabolites in blood and urine, and then also other aspects of comorbidities, insulin resistance, diabetes, and carotid atherosclerosis. Our prospective analysis will now include progression, incident diabetes, CVD events, and other comorbidities. So I think we now have a really well-established system to collect and understand the interrelationship between the gut microbiome, host and microbe metabolites, and non-AIDS comorbidities. We have gone on to look further to correlate the gut microbiota and host plasma tryptophan kynurenine metabolites. And in this case, we're actually able to show directly, here that these particular bacteria, as denoted here in the red box, do correlate now with the activity of the IDO activity, so that there are homologs to the tryptophan kynurenine metabolism related enzymes that these particular bacteria are able to make. And so this is now a more direct source of the bacteria, of these particular bacteria that are important in driving then this response. This was also shown in the original papers from Ivan Vukovic and Mike McCune, that the bacteria themselves are capable of making the IDO enzymes and contributing to the changes in tryptophan catabolism. And we're also now looking at another important pathway, the indole pathway. The indole pathway pathway is critical because this is the anti-inflammatory pathway for tryptophan catabolism. And so if you can drive tryptophan more to the indole pathway, you have more of an anti-inflammatory state. And we do find then that there is a reduction here in terms of the pathways for indole as well. So an increase in the tryptophan catabolism through the IDO, a reduction then in the indole pathway, leading to a more pro-inflammatory state that we think is directly driven through the host microbiome. What I'd like to do now is to move on to another story that we're developing in collaboration with Cécile Trombay's and Mohamed Alfar's lab at the University of Montreal. This I'm showing you in vignettes because these are all associated stories in CVD but tie together nicely in understanding pathogenesis. In this case, we're doing work on IL-32, looking at it as a novel player in cardiovascular disease. And what is known is IL-32 is involved in different inflammatory conditions, including with aging and cardiovascular disease. What has been shown previously in publications here is that IL-32 induces the expression of the metabolite indolamine 2,3-dioxygenase. So IL-32 gamma itself, and this is in tissue studies here, showing IL-32 and IDO expression from this paper in 2011 in Journal of Immunology, was able to show directly the link between IL-32 and IDO activity. So this now provides a link to what we were showing previously of an important role now potentially for IL-32 in cardiovascular disease. It's also known in vivo IL-32 is associated with IDO, ITL4 expression in macrophages and gut epithelial cells, so that there is the local expression then of the IDO pathway that could be having an effect, an outcome. We've also, again, with collaborations with Mohamed's group, and this was some of his work published in Science Reports just last year, showing that IL-32 does correlate with markers of disease progression. As I'll show you in a moment, IL-32 as a cytokine is much more complex. There are many isoforms, up to 5 or 6 isoforms of the molecule that leads to more of its complexity. It was shown in this original science paper that the increasing levels of IL-32 does correlate with increasing viral load and reductions in CD4. And as well, one can see increasing levels here as you move from HIV negatives to HIV positive treated individuals. It's known then that there is an IL-32 transcription and splicing to shorter isoforms, as is shown here, through gamma, delta, and alpha. And as I'll talk in a moment, we're now just developing the real-time PCR assays and the flow cytometric assays in collaboration with the group in Montreal to be able to measure these directly in our patient populations. So the hypothesis of this particular study, again, which is another recently funded grant application in aging, is that we hypothesize that there's a balance between the inflammatory and anti-inflammatory roles of IL-32. So this has both a pro and anti-inflammatory effect, and this can be linked with persistent inflammation and cardiovascular disease. The work that's being done currently is through the Canadian HIV and Aging Cohort Study. This is a large 2,000 patient study throughout the whole country of Canada that Cécile Tremblay is the PI on, and we've now nested our R01 study within this. We're also incorporating studies, because these are mostly men, we're incorporating studies from the WISE cohort that I showed you previously to also link with IL-32 changes, so that we'll be able to look at sex differences in our cardiovascular links in IL-32. And what one can see here is that based on age and aviremic men and women, that there's clearly an increase in both. But then when we look between men and women in age, that there's a significant drop in the IL-32 levels in the women and not in the men, so that there are, again, some significant differences, decreases with age in women but not men, and the decline of IL-32 in these aviremic women is not associated with the duration of HIV infection and duration of therapy. So this creates, again, more complex interaction of what's going on in terms of the HIV effect and the aging effect, and I think that's really one of the critical things we need to take into account in terms of our studies in aging and HIV is what are, in dissecting, what are the HIV, what are the aging studies. We've also shown recently is that there's correlations with the standard measures of the cardiovascular risk, including D-dimer, CRP, LDL, HDL. So that there's, again, the correlation here is with total IL-32, which doesn't represent all the different isoforms. So this is early data, and we also show a tendency for a negative correlation with coronary plaque. The expression of the individual isoforms also varies, and I'm not going to go into great detail on this, but there's higher levels of all IL-32 isoforms in HIV-infected individuals here, looking at across here of alpha, beta, gamma, delta, and epsilon. So we can now begin to look to sort out the effects of the different isoforms of IL-32 in our HIV populations. It's also important that the expression of these isoforms does correlate with atherosclerosis in our patient population. This is looking at total plaque volume here, showing, at least with the IL-32 data, delta, that is altered, and then here in epsilon goes up. So there are opposite effects of the of delta and the epsilon, looking at potential roles for both pro and anti-inflammatory effects of our different isoforms. And then overall, it's the IL-32 delta that correlates with coronary artery plaque volume. So it appears that this may be the most important of the pro-inflammatory isoforms. We're now sorting this out further in our studies and now looking at the ratios of delta with alpha, beta, and gamma, and showing again that there's a correlation across all of those different isoforms. So in conclusions for this part of the talk, the total pool of IL-32 remains higher in our HIV-positive patients on CART-treated and aging subjects compared to the general population. So there's both an effect of aging and HIV. In aviremic subjects, there's a sex-related difference, as I mentioned, for IL-32. We also know there's a tendency for a negative correlation between total IL-32 protein levels and total coronary artery plaque volume. So now we think that we have another new biomarker, and as I showed you earlier, we're now digging deeper in terms of the microbiome and the IDO pathways in these same patients. And we also know transcriptional levels of IL-32 delta, or its ratio, is positively and significantly correlates with higher plaque volume in terms of its potential role as a pro-inflammatory molecule, and we think then that may be also linked, as we're going to be doing further work of delving more into its role in the IDO pathway. So this whole study now is moving further. I might also add, this study is being done in collaboration with Nicolas Chermont's lab at the University of Montreal, and we're actually digging deeper in terms of the role of IL-32 and the latent reservoir. I think that's an important critical link that we don't see in many studies, is linking the idea of virus reservoir, comorbidities, and inflammation. And I think more studies can be done that way to understand that relationship of how these critical factors can drive important clinical outcomes. What I'd like to do then in the last part of around 10 minutes of my talk today is move on to another study that we're doing in collaboration with Ivana Pandria's and Krishna Pertree's lab at the University of Pittsburgh. This is now work moving on to study cardiovascular disease in the primate model, which gives us a better opportunity to manipulate the host to understand more about the effects of different factors. I brought up earlier the effects of lifestyle factors, and one of the things that we began discussions on a number of years ago was what effect does diet have on outcomes? As you know, the diet varies tremendously across the world. It affects our host microbiome, but we've really never in our human populations asked the critical question, what is the role of diet in our HIV outcomes, and how can we study that more effectively? So we set about to do this in the NHP models of pathogenic and non-pathogenic SIV infection. And in this case, we looked at the pigtailed macaque, which is the pathogenic host, where you find altered mucosal barrier, microbial translocation, and SIV comorbidities. We then compared this with the African green monkey non-pathogenic host, the natural host, where there's maintenance of mucosal barrier, lack of translocation, lack of chronic infection and comorbidities, and used the same virus to study now the host outcomes. In this case, we're interested in diet. And so we're now taking monkeys off their standard monkey chow and giving them high-fat diets. The amount of high-fat diet is they're getting 40% fat in their diet, and they're maintaining the same caloric intake as the non-high-fat diet animals on Purina monkey chow. This would be akin to going to the McDonald's for every day, for every meal, for a month. So I don't know how many of you could go to McDonald's every day and eat a lot of Big Macs or burgers here in Brazil, but that would really, you can see what the outcome will be here today in this study in our monkeys. What we've also done then is to try to understand this, and we're doing further. So first thing we did was look at the impact of the high-fat diet on the microbiome. One of the interesting features that we're now pursuing is that when we look at the actual diversity of alpha diversity here in our animals between AGM and PTMs, we see very clearly that there's a significant difference in the actual diversity of uninfected animals to begin with. So this raises some very interesting questions about how the endogenous host microbiome to begin with may affect the outcomes in our studies like this. And I think this again raises an important question as you're doing either animal model or human studies. How do you then control for some of these factors that may affect the outcomes of your various studies? We then go on to look at AGM and show a highly significant difference in the fat versus non-fat, both for the AGM and the PTM animals. We've gone on to look at the impact of high-fat data on the microbiome. I'm not going to go into great detail, but the major bacteria that were lost in our high-fat diet animals were those that were butyrate-producing bacteria. Now, this is critical because butyrate is a short-chain fatty acid that has prominent anti-inflammatory properties. It's one of the main mechanisms in the host, in both primates or humans, in maintaining gut barrier integrity. And when you lose these butyrate-producing bacteria, and we've published this in other studies, both in human and monkey models, you then lose gut barrier integrity. So this is, again, an important effect of high-fat diet, is losing your butyrate-producing bacteria, increasing the pro-inflammatory environment, increasing then activity of microbial translocation. We've also gone on in this study to show the high-fat diet are associated with biomarkers related to pathogenesis with our various bacteria at the genus level. And we've done an extensive look across all of these inflammatory cytokines, microbial translocation markers, other markers like hyaluronic acid, soluble CD14, and again can find very good correlations with the loss of the butyrate-producing bacteria and the increase of microbial translocation. We actually independently published a very similar study recently showing the same idea that when you lose butyrate-producing bacteria in humans, you increase levels of soluble CD14 in the systemic circulation in HIV patients. So there again is a good correlation, and we think now, how can we manipulate that? We're actually looking at how to reconstitute through the use of prebiotics in both our animal and human systems to try to bring back the butyrate-producing bacteria. The high-fat diet also alters the gut immune environment and activation status. And here we're just looking then at the macrophages in the tissue, looking at CD80 and 86. Just as an example, the control animals here of the pigtails, macaques, and then the high-fat diet. So you can see no effect on SIV infection, but the animals that are SIV infected and on the high-fat diet get this very significant impact in macrophage activation. We also have begun to look at metabolism effects as well, which I'll talk about in a moment. We can see very clearly that there's a high impact on the gut mucosal inflammation here, and this is looking at myeloperoxidase, again, in pathogenic pigtailed macaque model, so that we can find then a high level here at necropsy of the MPO or inflammatory mediators in our gut tissue. So there's a lot of inflammation going on in the animals themselves. As I mentioned, we're also interested in immunometabolism and have begun to look at both at the T cell and the monocyte levels of metabolic effects. What effect does high-fat diet have on immunometabolism? We've begun to use the Seahorse analyzer that gives us a way of looking at mitochondrial stress effects at the single cell level in our animal peripheral blood mononuclear cell populations. And what we were able to show very clearly in these animals is that there's an increase in the extracellular ECAR activity, which is the glycolytic activity in the animals. This is pre and post PTM fed high-fat diet. So that there's now a driver of the glycolytic over the oxidative phosphorylated pathway of our both T cell and monocyte populations in the animals. What this means then that these cells are driven more to an inflammatory state and to a higher activation level. We've, in other independent studies in the WISE cohort, have also done similar work and shown that this high level of glycolytic state with GLUT1 expression is associated with cardiovascular disease. So again, we think that this is a very consistent finding that we can use to study the role of cardiovascular disease in both the experimental macaque model and in humans to link with metabolic changes and outcomes related to cardiovascular disease. What we've also done is to look at high-fat diet, increasing the severity of cardiovascular lesions. And in this case, we're showing very clearly in our animals that died, this again is the PTMs, that there are foamy cells, thrombus formation, thickened intermedia, so that there is, again, evidence of severity of cardiovascular lesions. So there's direct effect now of the high-fat diet. This is in SIV-infected animals. In the SIV-infected animals, not on the high-fat diet, we do not see these same pathologic changes. So there's clearly an impact on our host that's independent of the SIV, linking it directly to the diet. And we think this, again, creates another model for us to be able to study in the SIV-infected host of cardiovascular disease outcomes. We're looking at other pathologic effects as well now in these hosts. One of the most significant, though, effects of this high-fat diet in our animals now has been survival. And although this was not significant, it was marginally significant. These are small. This was a pilot study of 5 high-fat diet PTMs and 10 control animals. But you can see that in all of the fat diet animals, you had 100% mortality, whereas it was only, you had, again, the percent survival was 60% here, or 40% mortality in the normal chow-fed SIV-infected animals. So there is, again, a significant effect of putting these animals on high-fat diet on their mortality. Interestingly enough, in the non-pathogenic host, there was actually 1 death related to this, to the same kind of pathology. And as you can see here, in these AGMs, those on chow, no deaths, but there was 1 death in the high-fat diet fed animal, and again showed similar cardiovascular and other lesions. We have extensive pathology on that as well from Dr. Pendria's lab. So we think, and again, this is a significant effect on the outcome, something we need to incorporate and understand more about in terms of our integration of our understanding of the role of diet in HIV pathogenesis, and especially, as I just mentioned to you, related to cardiovascular disease. So in conclusions then, I'd just like to say that diet may be a critical contributor to the residual immune activation and inflammation in our own subjects. As we know, people as they age do gain weight, higher BMIs. Many of our cohorts that we're studying in our HIV positive women in the US, very high BMI levels, and we're now looking at the differences between our men and women cohorts, where we know there's a very different epidemic in women in the South in the US. I don't know here in Brazil, and maybe during our discussion we could talk more about differences among populations even in Brazil and how diet across Brazil might affect the outcomes of the host, as we're seeing now in some of our US cohorts, and also the role in the primate models. We know again that the diet affects metabolic changes. I've shown you some of those that we're now looking at by immunometabolism and seahorse analysis, and that we're also, and I didn't show you the data, but significant liver damage occurs in the host here as well. We know that there's systemic inflammation that leads to abnormal fat distribution as well, and deposition that we saw in these animals. And eventually then we also have abnormal coagulation markers leading to some of the cardiovascular outcomes. So we think this is a great model to be studying the interactions then of all the various host factors and organs, as is said here, that the diet can impact the gut, liver, and cardiovascular system axis and promote comorbidities and accelerate potentially disease progression in our host. So we think now this provides another model in cooperation with some of the other novel biomarkers I was just describing to you that might be able to integrate in our better understanding of how inflammation and aging intersect in the HIV-infected host. So I'll just thank my collaborators here on the last slide. As I mentioned, the University of Montreal, Cécile Tremblay and Mohamed Alfar for all the work that is ongoing for IL-32. This is funded under a recently funded R01 under the National Institutes of Aging. Our work on the at Einstein and also with Johns Hopkins, SUNY Downstate is being done in collaboration on the tryptophan metabolism. We're doing our primate work here with the University of Pittsburgh group on our fat diet outcomes. And then finally, Peter Hunt, who've had a lot of long discussions about the role of aging, HIV comorbidities. I'd like to thank you for your attention and happy to take any questions.
And I open to the floor for questions. Mike?
So, Alan, your monkeys who fed this high-fat diet and who had SIV infection and who died, did they die of cardiovascular complications or did they die of opportunistic infections?
They're dying of cardiovascular disease and not OIs directly, Michael. Yeah.
All right. Cool. Thank you.
So it looks more relating to the diet and not the virus itself. And it could be the virus, yeah. I mean, we're...
So I think one could propose that that there are bidirectional relationships.
Sure.
If you look carefully at the lipidome of these animals, there are more than 1,000 different lipid molecules, some of which have anti-inflammatory effects, and some are highly pro-inflammatory and immunosuppressive. So I'm not saying that they did die of heart disease, but they may have an more aggressive immune phenotype as well.
Yeah, I think we still have all the samples, and I think this is now a new grant, so this was just the preliminary data to fund a newly funded grant that will be moving forward and looking at those questions.
Hello, thank you so much for your excellent talk. So I am just curious to know that for the cardiovascular disease, if someone thinking for the treatment, which one is important, like glycolytic pathway or oxidative phosphorylation?
Yeah, well, we're now doing that. So the question was looking at the immunometabolism in treatment. We're just now doing those studies. What we find is we're doing the sequential studies right now to see what happens in terms of reversals. The more resting state is the oxidative state of the immune system. And it's interesting when you start to look at that interaction, it's very complex in terms of different cell populations like T regulatory cells, Th17 cells. All of them have different glycolytic versus oxidative phosphorylated state. So we're just now looking at the bulk CD4s, CD8s, and monocytes. What we need to do next is to drill down to the separate cell populations and understand what's going on in terms of their state. But what we don't see, at least overall, by surface markers is on therapy, they still seem to have a higher level of glycolytic activity, at least by GLUT1, and we can measure that functionally by a flow mass measure of NDVG, which is a measure of glycolytic activity. So we do see at least that. We haven't done yet the seahorse analysis on the longitudinal samples.
Good morning, Professor. Here. How could the tryptophan metabolism management affect the ILC population in the gut once ILC is activating those cells, and what may lead to bowel diseases? So how could bowel diseases be avoided if ILC levels keep higher?
Okay. So the question is about innate lymphoid cells in the gut, and we haven't yet looked. We have other data in collaboration with Carol Wilson's group and Ed Barker. We have published work in Steph Dillon on changes in ILC populations with relationship to ILC3s becoming more pro-inflammatory. They are normally important producing IL-17 and IL-22, and they're now being turned on to make interferon gamma. We haven't looked directly at the role of the microbiome interacting or tryptophan catabolism. That's something that we're looking at in another grant going forward on our ILC work, is to try to determine the interaction of the microbiome more directly on the ILC populations and the host metabolic or bacterial metabolic pathways.
Alan, you seem to be on a fat crusade. And I remember that we used to all worry enormously about fats, and now all of a sudden the diet folks are shrieking about carbohydrates. Can you speculate and talk a little bit about the relative importance of carbohydrates versus fats in terms of the morbidity of your animals and the morbidity of your friends?
That's a great question, Michael. Okay. That's actually a great question. We control for the carbohydrate levels, at least in these initial studies, but the next studies are actually going to look at the effects of carbohydrates. So we're going to independently evaluate in the grant that we're now just starting to ask about carbohydrate independent of fats and seeing exactly what happens in terms of the sugar input and how that might be. We're redesigning all the diets now to try. This was a very high level, 40, as I mentioned, 40%. So we looked at the extremes in our initial study, but we think now we can go further, refine the diets. There are groups at Wake Forest that have been actually defining all these different diets for monkeys based on fat and sugar contents. But I think that's an important and very relevant question that we're now pursuing. Thank you.
In the first part of your talk, when you showed us the imbalance between tryptophan, IDO, microbiota in HIV patients, you mentioned several times possible targets. So if you would think in a possibility of interfering this and reestablish the balance, where would you interfere? Why?
Well, as I said, IDO inhibitors we're starting trials at our own university on IDO inhibitors in combination with checkpoint inhibitors. It's probably the biggest area in a lot of new cancer areas using IDO inhibitors. There's been a lot of interest in small molecules, but they're moved into the cancer field, and we're now trying to see could that be also applicable. There has been data, and I will say early data from Vefa Franchini and Mike McCune, where they treated with 1-methyltryptophan, which is the inhibitor that David Munn originally and is continuing to work on with regard to cancer therapy. They did show some untoward effects in those animals in developing diabetes, so they didn't do well. They actually ended up with significant mortality with at least the 1-methyltryptophan treatment to try to inhibit the IDO pathway. So I think now we need to look at other newer small molecules to try to see if we can reverse the effects of that. We're also looking not only in the cardiovascular, but also in the neurocognitive outcomes, and there's a whole range of areas there. What also modifies the IDO pathways in the tryptophan pathways is also related to melatonin and serotonin levels. So we're looking at whether or not we can move along different pathways, and also with indoles. There are and we're working with collaborators at Emory University who actually have new molecules to drive from tryptophan down the indole anti-inflammatory as opposed to down the IDO pathway. So we're going to explore a number of potential opportunities there.
Yeah, I have seen some of these trials on IDO, small molecules interfering with IDO metabolism. They're quite difficult to control, actually. It's a very complex metabolism. Why would you not try to interfere in microbiota in this case?
Oh. No, I think that I also would say we could try to do and alter the microbiome to affect. You saw there are effects in VisBiome studies that came out. Ettori's work group in Italy using VisBiome did show some effects where they started to look at that. So there is the possibility as well to look more broadly with the probiotics. I should mention, just completed a VisBiome study in the ACTG and are looking at what that did to the tryptophan catabolites. But I should mention, you're absolutely right. The microbiome and manipulating it is another target, much less toxic and much easier perhaps to intervene.
Any other question?
About how a fat high diet could influence in heart disease. we can assume that people who have high levels of cholesterol for genetic reasons could have the same risk?
Well, just asking, yeah. If you have genetically high levels of cholesterol, could you have similar risk? Of course you could. So I think, again, was brought up by Dr. Letterman earlier on, there's a high degree of complexity in our lipids. And now that we're doing much more on lipidomics and understanding the pro or anti-inflammatory role of lipids, I think we need to sort of drill down further and see what is it that we can directly modify. But I think it just raises the idea, and also how to define the best diet, I think, is always going to be a critical question, and determining its role and effect on the microbiome is where we're going to be focusing our efforts.
Last question.
Again. So, do you think microbiota transfer from the gut of a healthy individual to a HIV subject, or the monkey model, could have some impact in the prognosis of disease?
Yeah, that's a great question. Fecal transplants have already begun in the HIV and have been published. Mas Samsuk's group from UCSF has already published, in collaboration with Ivan Vukovic from the NIH, on the use of normal donors in HIV, showing very transient effects on the microbiome, very different than with the C. diff experiments of treating patients with Clostridium difficile. One of the questions is whether or not you have to actually remove the host microbiome, as C. diff will remove most of the host bacteria. So you've already sort of created the fertile soil for the microbiome fecal transplant. And so that's what they're now looking at, using rifaximin in the next stage. And we're also looking at fecal transplants in our HIV populations in the ACTG. One of the interesting questions you raised, which I showed you earlier, is that there's a difference in the microbiome between pigtails and African greens, significant differences even at just regular in the animals living in colonies. So we're now starting to do and set up experiments to do fecal microbiome transplants among the different monkey models to see how that affects. And then one final word is, because we're going to hear more about vaccines, at our last microbiome meeting, Dr. Hazuda presented data from Merck on the vaccine responses that they can show, and are doing fecal transplants in older monkeys from younger monkeys to see if they can now impact the actual vaccine response, which she's shown are altered in their older versus younger monkeys. So that time will tell from those studies at Merck to see if there's an effect of the microbiome on vaccines as well.
Okay, thank you very much, Alan.